Good day, class. So for today, in our biomolecules, this will be a recorded discussion regarding proteins. So proteins has a monomers, or the building blocks of proteins is your amino acids. So amino acids have this kind of structure. So as you can see, it has a amino group and a carboxyl group, okay, and an oxygen as its central atom. So again, the elements in your amino acids are carbon, hydrogen, oxygen, and nitrogen. So as you can see, it has its amino group and carboxyl group. So being an amino acid, okay, as the term implies, amino coming from amino group and acid coming from carboxyl group, because this will be your carboxylic acids. Okay, that's why it got its name amino acid. So your amino acid can be attached to different side chains depending on what type of amino acid it is. So later on we'll be discussing that. So again, your amino acid is the building blocks of protein. So this is the smallest units for your proteins. So what are the characteristics of your amino acid? So first, the fundamental subunits of carbon, hydrogen, oxygen, and nitrogen. So also, your amino acids are optically active due to the presence of your chiral carbon, because again, your carbon forms four covalent bonds, correct? So because of this, it is characterized as an optically active atom or molecule, I should say. So when we say optically active, it has the ability to rotate plane polarized light in a polarimeter. So it can either be dextrorotatory or levorotatory. So when we say levorotatory, it has a positive sign wherein plane polarized light shifted to the right. Okay, so it's to the right. But compared to your levorotatory, the plane polarized light shifted to the left. Okay, so we're talking about here the rotation, okay, of your light. Also, your amino acids are amphoteric. What does it mean when we say amphoteric? It can act both as an acid and a base because of the presence of your amino group and the presence of your carboxyl group. When we say acid again, these are also known as proton donors, while base are proton acceptors. Okay, so your COOH is your proton donor or your acid group, and this is your amino, which is your proton acceptor, and this gives the basic property of your amino acid. Also, your amino acid is a dipolar ion, wherein in other terms, it should be Zwitter ion. Okay, so it possesses both positive and negative ion. So this is also actually with your amino group in your carboxyl group. So acidity is due to the electron delocalization. So the OH bond in COOH, so this one, weakened, so it is readily or easily given off or loose or lost as a proton and is accepted by the lone pair of your NH3. This is NH2, okay? So it becomes NH3, okay, as it accepts proton. So that's your Zwitter ion. So your amino acid as a Zwitter ion. Next, we have here your isoelectric point. So this is a pH, okay, a pH value at which Zwitter ion state exists at a maximum. So there is a specific pH where your amino acid achieve being a Zwitter ion or a dipolar ion. Okay, so amino acids and proteins assume a zero net charge when they have a neutral charge. Okay, amino acids and proteins are least soluble. So meaning your amino acids form precipitate. Okay, when subjected to acids or into high pH or low pH. So no integration in the electric field because they don't have charges. Okay, they are neutral already, so there is no integration in an electric field. So um, each amino acid has a specific isoelectric point. Okay, so the when the amino acid has an above pH compared to its isoelectric point, it favors an anion state. But if it's below the isoelectric point, the pH is below the isoelectric point, it favors cationic state already. Okay, so for example, we have here the different isoelectric point of amino acids. So we have here your aspartate 2.8, glutamate 3.2, serine 5.7, valine 6, alanine 6.1, lysine 9.7, and arginine 10.8. Okay, so those are the isoelectric point of some amino acids. So, what is the importance of isoelectric point? It is used in buffer system. When we say buffer again, it is the ability to resist the change in pH. Also for separation and identification process because as I told you, each amino acid has its distinct isoelectric point and also shows structural changes in globular proteins. So this explains the structure of biologic reactions because some of each proteins undergo different biologic reactions. So depending on their structure. Okay? So we have here a problem. So given serine, which has a isoelectric point of 5.7, what structure dominates at? So as you can see, if your serine is subjected uh, to a pH of 5.7, it becomes a Zwitter ion. So it has both negative and positive charge. So this is its um, isoelectric pH, right? So 5.7. But if your if your serine is subjected to a lower pH, so this is 4.2, okay, your serine will be in cationic state. Okay, it becomes positive already. But if it is subjected to higher pH than your 5.7, it's 8.5 here. So your serine will be in anionic state. Okay, it already has a negative sign here. So that's anionic. But if you say cationic, it has a positive charge. But anionic has a negative charge. So that's the characteristic of your amino acids with isoelectric point. So these are the different um, amino acids. So we have 20 amino acids all in all. We have your alanine, arginine, aspartic acid, aspargine, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. So these are the 20 amino acids. So each amino acid is named depending on the 3-letter code and 1-letter code. So as you can see here, it has its 3-letter code and 1-letter code corresponds to each amino acid. So with this, we have here also your 21st amino acid. So we have your selenocysteine. But usually the selenocysteine is not used or it's not present during the coding of genetic informations in your nucleic acids. Okay, because again your glutamine will be the final product of um, what do you call this, translation, okay, of your DNAs from DNA to RNA, DNA replication to RNA transcription, then translation. Your RNA becomes amino acid. Okay, so it doesn't. Your selenocysteine is not present there. Only this 20, okay, is present during translation. Okay, because each codon in the um, Each codon of RNA in transcription will be translated during translation, and it will become an amino acid. So what are the different classifications of your amino acid? So first, it depends on the structural classification. Okay, first, it depends on the aliphatic side chain that is present. Okay, when we say aliphatic, again, it's a hydrocarbon. Okay, it has carbon and hydrogen um, atoms in the group. So as you can see here, we have your glycine. It has H, alanine, CH3, valine. As you can see, we have CH3, CH3, okay? Also have your leucine, methionine, and isoleucine. So as you can see, all have hydrocarbons attached to it. Next, um, with side chains containing hydroxyl groups or OH groups. So we have here your serine, threonine, and tyrosine. So as you can see, here's the OH group, here's the OH group attached, and here's the OH group attached to each amino acid. Because again, this is the general structure of your amino acid, right? Okay. Next, with side chains containing sulfur. So we have here your methionine and cysteine. So we have here sulfur and sulfur attached to it. So this again is your amino acid. So each a group that is attached, it contains sulfur. And side chains containing acidic groups or their amides. So again, carboxylic group is COOH, right? Or COO. So aspartic acid, this is your COO, COO2 aspargine, glutamic acid, COO, and glutamine CO here. Okay. Next, with side chains containing basic group. So basic, it has NH3 or NH2. So as you can see here, we have your lysine, NH3 here, arginine, and histidine. Okay, so it has NH2 or NH3. And with containing aromatic rings. So as you can see, we have here your phenylalanine, tyrosine, and tryptophan. It has aromatic rings. And we also have your amino acid. So as you can see here, your um, group that is attached or a radical that is attached, it was attached to both carbon and the amino group of the amino acid. That's why it's called amino acid. Okay, so this is your proline. As you can see, this is the group of your amino acid, right? So this is the amino acid structure. So it is attached to um, hydrocarbons in both ends. So that's your amino acid, which is protein. Also, your amino acids can be classified according to polarity. When we say again polarity, it is the presence of um, more electronegative element in the structure. Correct? So that it can be considered polar. It can form hydrogen bonding. Okay? So we have polar and non-polar amino acids. So under polar, we have your aspartic acid, glutamic acid, arginine, lysine, histidine, aspargine, glutamine, serine, threonine, and tyrosine. Under nonpolar, we have your alanine, glycine, valine, leucine, isoleucine, proline, phenylalanine, methionine, tryptophan, and cysteine. Also, we can classify your amino acids depending on their nutritional importance. So we have here your essential and non-essential. So when we say essential, these are amino acids that can be synthesized by the body. That's why it is needed to be taken regularly. Okay, or it must be included in the diet so that it will be present in our body. But when we say non-essential, these are already present in the body. So there's no need to take food or vitamins that has this um, amino acids already, or because they are already present in our body. So under essential, we have your arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. But under non-essential, we have your alanine, asparagine, aspartate, cysteine, glutamate, glutamine, glycine, proline, serine, and tyrosine. So again, when we say essential, these are indispensable amino acids. So those which cannot be synthesized in the body that must be present or provided in the diet. So primary concern with growth and maintenance of life. But when we say non-essential, these are dispensable amino acids, those which can be produced in the body. So those are your essential and non-essential amino acids. So again, your amino acids are the um, monomers of your proteins, right? So a chain of amino acid, it will be your polypeptide because amino acid plus a bonded to an amino acid, it is a peptide, okay? It is bonded by a peptide linkage. Okay, so a chain of amino acid is your polypeptide. So a multiple chain of polypeptide will make up your proteins. So, what are the now let's go to the properties of your proteins. A while ago it's amino acid, now it's proteins. Usually, the characteristics of your amino acid is just the same, okay, of your proteins. It's just that your proteins are much bigger molecules compared to amino acids, right? So, generally, the physical properties of your proteins generally are colorless, odorless, tasteless, okay, but except for one protein, which is the protein hydrolycates, which is bitter. Also, acids, bases, and salt solutions varied solubility in solvent. It also forms colloidal dispersion, which do not pass through membranes. So your proteins are big molecules that can't pass through membranes. That's why it forms colloidal dispersion. And also low molecular weights of proteins are soluble in water. When we say low molecular weights, again, these are proteins that has lesser amount or lesser molecule in the structure, while high molecular weights or macromolecules, so these are big molecules already, are totally insoluble in water. So what are the chemical properties of your proteins? Also, they are amphoteric or ampholyte. Again, they possess both positive and negative ion, meaning they are Zwitter ions, right? So, due to the presence of COOH, which is an acid, it is a proton donor. It can readily gives off protons, while NH2 is a base wherein it accepts proton. Okay, because again, the general structure for amino acid, it has an amino group plus a carboxyl group, correct? So, because of this, they are generally good buffers. Okay, so they can resist the change in pH. Also, your proteins are precipitated by the different solutions. We have your strong acid solutions, salt of heavy metals such as your copper sulfate, this is lead acetate, okay, and we also have your mercury chloride. Also forms precipitate with alkaloidal reagents. So these reagents came from plants. And also we have here the property of salting out or high concentration of neutral salt solution because there is a formation of precipitate, okay, because of the high concentration of neutral salt. Next, so we have here your denaturation of proteins. When we say denaturation of proteins, this is the destruction of the secondary, tertiary, and quaternary structure of your proteins that leads to its physical, chemical, and biological characteristics. Okay, so the meaning this is the destruction of structures because we have different organizational structures of your proteins. but the primary ones is not being denatured. Okay, so what are the different factors that affects denaturation of protein? So first, physical factors, we have your heat, ultraviolet rays, and high pressure. Under chemicals, we have your acids, bases, and organic solvents. So the changes, okay, that affects your proteins, for physical change, it increase viscosity, okay? It becomes viscous. While in chemical change, decreases solubility by enzyme. Also, your proteins are subjected to hydrolysis. When we say hydrolysis, this is the breaking down, okay, of proteins into smaller pieces with the reaction of water. Okay, this can also happen by acid by reacting it to acid, bases, and some protein splitting enzymes. Also, it's the opposite of peptide formation because again, it's a catabolic reaction. Your hydrolysis is a catabolic reaction, meaning you're breaking proteins into its smaller units. And water is added to form back the original amino acid. So as you can see here in the example, your proteins plus water reacts to proteases, okay, and water, it becomes peptones. So your proteases are enzymes that are reacting to your proteins to broken down into smaller size, which is your peptones, okay. And your peptones, again, subjective to hydrolysis, it becomes polypeptide. Then hydrolysis again becomes dipeptide, and then peptides, then amino acid. Okay? So that's a catabolic reaction. So that's your hydrolysis. Now let's go to structural organization of protein. So this is what you've heard a while ago regarding the denaturation of proteins, because only secondary, tertiary, and quaternary are the ones being denatured. Okay? So first, let's go to your primary structure. So these are linear arrangement of amino acids in a polypeptide chain joined together, excuse me, joined together by means of peptide linkages. So this is a straight chain, okay, of amino acid bonded to each other by means of peptide linkages. So, what are the characteristics of your peptide linkages? So, first, it is a strong bond, okay, due to the electron delocalization towards the carbonyl group, which is your COOH. Okay, so it forms strong bond to the amino group of the other amino acid through electron delocalization. Okay? And also exhibits, geometric isomerism. When we say isomerism again, um, it has same molecular formula but different structural formula. Okay? So we have cis isomers. When we say cis isomers, both the functional groups attached is both located above or both located below, or both located on the left side or both located on the right side. But when we say trans isomer, um, one group is attached to one side and the other one is on the opposite side. So such as this, one is located above and one is below, or one is from left and one is located to the right. So that's your trans isomer. Okay? So due to the restricted rotation in the double bond, group attached to carbon and nitrogen may be projected on the same side. Next. Now let's go to your secondary structures. It involves folding of polypeptide chains due to hydrogen bonding of peptide bonds. A while ago, from primary structure, your amino acid is bonded through peptide linkages. But with this, due to the hydrogen bonding, there is a folding already that happens. So it forms sometimes a pleated sheet in a alpha helix. Okay, so your secondary protein structure, hydrogen bonding of the peptide backbone causes an amino acid to fold into repeating pattern. So it's either pleated sheet and alpha helix. So we have two types of secondary structure. First, alpha helix. When we say alpha helix, it is formed due to intramolecular hydrogen bonding. When we say intramolecular, the bonding happens within the molecule or within the peptide itself. But when we say pleated sheet, due to the intermolecular hydrogen bonding. So intermolecular meaning between different peptides. Okay, hydrogen bonding happens between different peptides. Compared to alpha helix, it is bonding, hydrogen bonding happens within the same molecule or same peptide. Okay, so your hydrogen bonding is a weak band due to a electron weak attraction between alpha and hydrogen and lone pairs of the atom. So your hydrogen bond is represented by dotted lines. Okay? So that's your hydrogen bonding. Next, let's go to your tertiary structure. So this is the unfolding of polypeptide chain due to the formation of bond like hydrogen bond, covalent bond, and disulfide bond. So there are so many bonds already that happens in your peptide chains. Okay, so it is a three-dimensional folding pattern of a protein due to side chain interaction. So as you can see, we have here a pleated sheet already. This one is a pleated sheet and your alpha helix already. Okay, so um it was mixed already and was there's already also another folding three-dimensional folding pattern due to the side chains interaction already. Then now let's go to your quaternary structure. So interaction of subunits. Okay, so we have two already here, two or more amino acid. So proteins consisting of more than one amino acid chain. So it involves polypeptide chain tightly woven to each other. So that's the structure of your quaternary structure. So now let's go to the pathologic side of your proteins. Okay, so we have here malnutrition. When we say malnutrition, it is caused by the deficiency or exists in one or more essential nutrients in the diet. So it is characterized by a wide array of health problems, including weight loss, stunt growth, weakness, resistance to infection, and impairment of intellect. And a severe cases of this may lead to death. So again, when we say malnutrition, it can be an excess or an deficiency of a certain nutrient. Okay. But so when we say protein malnutrition, this is due to the food people eat which are low in protein. Sometimes there's no protein at all and the body is not able and also the body cannot be does not synthesize protein to produce energy. Okay? So what are the different two cases, okay, two disease associated with your protein malnutrition? So we have here your kwashiorkor and your marasmus. So kwashiorkor is characterized by swelling of legs, okay, or edema, sparse hair, moon face with little interest in surroundings, flaky appearance of skin, swollen abdomen, thin muscles or but fat in present. So this is the a child having a kwashiorkor. But when we say marasmus, it has its normal hair, old man or wizened appearance, thin limbs, and little muscle or fat, and underweight and very underweight body. So these two are related to protein malnutrition. So this is the deficiency already, okay? Meaning there's no less protein in the body. Also, we have here sickle cell disease. So also known as your drepanocytosis. So actually, this is not a malnutrition, but a mutation. Because a while ago, if you remember, your proteins, okay, are translated from your RNA, okay? So they represent codons and they represent proteins with that. So here, because of those translation, there is already, what happened here is a mutation. Okay? Point mutation wherein there is one type of amino acid, or sorry, one type of amino acid was incorrect. Okay? So again, your sickle cell disease is a hereditary blood disease caused by a deficiency in protein globulin found in RBC. So it's characterized by the production of an abnormal type of hemoglobin. So it is a sickle cell hemoglobin in the RBC. When we say sickles, it is sickle cell because it was shaped like a sickle. Okay? So your sickle cell hemoglobin becomes insoluble when blood is deprived of oxygen, and precipitated, forming elongated crystals that distort the blood cell into the characteristic of a sickle cell. Okay, so because of this, when you have sickle cell hemoglobins, it can't flow uh, normally in the veins, okay, or into the bloodstreams. That's why sometimes it is stuck to a certain place, and um, it can form clogs, okay, in your veins. And also, this type of hemoglobin are less can't carry the right amount of oxygen that is needed by the body. So that's why it is deprived of oxygen because of its shape, okay. So that's your sickle cell disease. So as you can see here, this is the normal red blood cells. Okay? So in your sorry. So your DNA, uh as you can see here, there is a mutation in this part. Okay? Here. So your adenine here was mutated to thymine. Okay? That's why during translation it becomes adenine. So the normal should be glutamine. Okay, when it was translated, it becomes valine already. So this is a mutant protein already. So that's why this caused the sickle cell hemoglobin with people who have sickle cell disease. So now let's go to your classification of proteins. Okay, so we're done with classification of amino acids. Now, classification of your proteins. So your proteins can be classified depending on their different categories. So we have here functional classifications of your proteins. So we have here structural proteins. So these are your example of this are your collagen. So your collagen is present in your bones, right? That's why it's considered as a structural proteins. We also have here your storage proteins. Example of this are your ovalbumin and glutein. We also have here your catalytic proteins. So these are your enzymes because again your enzymes are protein in nature. Okay. We also have here your contractile proteins. We have here your actin and myosin which are present in your muscles. We also have here your regulatory proteins. So these are your hormones. Okay, so example, insulin and growth hormones. We also have here your receptor proteins. So these are the ones that receive or act as a receptor for hormones or for your regulatory proteins. We also have your genetic proteins. Example of this are your histones. And we also have your defense proteins. Okay, so example of this are your immunoglobulins. And we also have here your transport proteins. Examples of this are your albumin. So when we say transport proteins, because again, your proteins are big molecules, right? So it can't enter the cell membrane of the cell. So your transport proteins are located from the in the cell membranes of your cell. Okay. And the ones that transport the proteins outside to going inside the cell. Also, once inside, when we transport it outside the cell, it also needed transport proteins. Okay, so it can pass through the cell membrane. Also, according to nature and solubility. So we have here your simple proteins, conjugate proteins, and your derived protein. So when we say simple proteins, when subjected to hydrolysis, it only yields amino acid and alcohol. But when we say conjugate proteins, when subjected to hydrolysis, it produces amino acid in a prosthetic groups. When we say prosthetic groups, these are non-protein group, okay, that's attached to your proteins. And we also have your derived proteins. So these are when subjected to hydrolysis, it yields simple proteins and conjugate proteins. So under simple proteins, again, those that yields only amino acids on hydrolysis, amino acids and alcohols, okay? So examples of this are your albumin. So these are soluble in water and coagulated by heat, meaning when you are subjected it to heat, it becomes viscous. Okay, so example of your albumins, we have your ovalbumin from egg white, lactalbumin from milk, myogen from muscles, serum albumin from blood, and also we have here your legumulin from your plants. We also have globulin, so these are insoluble in water but soluble in dilute solutions. So coagulated by heat. So examples, we have your ovoglobulin from egg white, serum globulin from blood, lactoglobulin from milk. We also have from plants. So these are your legumin from peas and legumes, arakin from peanuts, pheidolin from beans, and amnin from almonds. Next, we have your glutelins. So these are insoluble in water in neutral solutions, but soluble in dilute alkalis and acid. Also, they are coagulated by heat and only come from plants. Examples, we have your glutenin from wheat and orizinin from rice. Next, we have your prolamins. So, soluble in 70% to 80% alcohol, but insoluble in water or absolute alcohol. It is found in plant seeds. So, examples of this are your zein, which is from corn, gliadin from wheat. Then we also have albuminoids. So these are insoluble in all solvents mentioned, least soluble among all simple proteins, and comes from only from animals. So it is called also as scleroproteins. So examples of this are your keratin, found in your hair, collagen in your bones, elastin on tendons, and fibrin and silkworm. So those are your simple proteins. We also have others. So we have histones, soluble in water in dilute acids, but insoluble in dilute ammonium oxide, hydroxide, I should say. Not and also not coagulated by heat. So examples of this are your globulin from hemoglobin, thymus histone from thymus gland, and gadus from codfish, and also scrombone from mackerels. And we also have your protamines. So this is the simplest in structure of all proteins. They are soluble in water, in dilute acids, and dilute ammonium hydroxide. And they are not coagulated by heat. Example of this are your salmine from salmon, styrene from sperm of fish, scombrine, from mackerel, cyprinine from carp, and cyprine from herring. So as you can see, these are all from fishes. So that's are your simple proteins. Now let's go to your conjugate proteins. So those on hydrolysis will yield a non-protein substances or called your prosthetic groups in addition to amino acids. So examples or under this we have your nucleoproteins. So proteins linked with nucleic acid. So the prosthetic groups will be your nucleic acids. So found in nuclei of all living cells. Example of this are your nucleoprotamines. So nucleic acid plus protamine when subjected to hydrolysis. We also have your nucleohistones. When subjected to hydrolysis, it yields nucleic acid and histones. We also have your glycoprotein. So from the word implies glyco, meaning the prosthetic group is carbohydrates. So this proteins link with carbohydrates. So the enzymes that is present during hydrolysis is your ptyalin, okay, or your salivary amylase. So example of this are your mucin, which is present in your saliva. So it is 99% water in 1% mucin, which makes our saliva viscous or lubricates or moisten food. Okay, so this is a glycoprotein. Then we have your mucoids from tendons and cartilages. Also oviomucoids from egg white, seromucoid from blood, and urine mucoid from urine of animals. Then we have your phosphoproteins. So phospho meaning it has a phosphoric acid or phosphorus atom or molecule. Phosphorus containing molecule, I should say. So example of this are your casein from milk and vitelline from egg yolks. We also have your chromoproteins. So proteins linked with color producing group from the word chromo. So high color producing group. So we also have here your hemoglobin that gives color to your blood, melanin to your hair, cytochrome, and flavor proteins. Next, we have your lipoprotein. So from the word lipo meaning fatty acids or lipids, right? Proteins linked to fatty acids. So examples of this are your fibrinogen found in your blood and proteins in egg yolk, milk, cell, nuclei, and cell membrane. So that's your conjugate proteins. Now let's go to your derived proteins. So, substance produced by the action of heat, acid or alkalizer enzymes on simple and conjugate proteins. So, this group includes all hydrolytic products of the original proteins. So, first we have your primary derived proteins. So, these are derivatives of proteins in which the size of protein molecules is not materially altered, okay, or not changed. So, example of this are proteins. So, these are insoluble in water. The first product produced from the action of acid, enzyme, or water on proteins. Next, we have your metaproteins. So, these are insoluble in water, but soluble in dilute alkalis and acid. So, produced by further action of acid or alkali on protein. So, this will be the next two proteins. Okay, so proteins first, then metaproteins will be produced. Then we have your coagulated proteins. So these are insoluble proteins because products produced by the action of heat or alcohol on proteins. So when your proteins are subjected to heat and alcohols, it will produce coagulated proteins. Okay, meaning they are super viscous already. Next, we have your secondary derived proteins. So these are derivatives of proteins in which definite hydrolysis has taken place. So the molecules are smaller than those of the original protein. So examples of this are your proteoses. So these are already the smallest um, form of your proteins already, or your very near to amino acid already. Okay, so your proteoses are soluble in water, coagulated by heat, and precipitated by saturating with, with solution, uh, saturating their solution with ammonium sulfate. Then next we have your peptones. So properties similar with those proteoses, but are not precipitated by ammonium sulfate. Then we have your peptides. So a combination of two or more amino acid and are the simplest derived protein molecule. They are known as dipeptides, tripeptides, and polypeptides. So these are chain of peptide bonds, correct? Depending upon the number of amino acid in the molecule. So those are your secondary derived proteins. So next, we have here the different tests for proteins. So as you can see, we have the name of your test and what is the test for, then the positive result is given. So for Ninhydid test, which is a test for alpha amino acids. So the positive result would be Robimon's blue. Okay, so this will be positive to all amino acid except for proline. Then we have your shift test. Okay, these are tests for NH2 group, amino group of lysine. So it yields a positive result of pink violet. We also have your Sagaguchi's test for guanidine group of arginine. So the positive result would be red orange. Then we have your Milanacis test. These are tests for tyrosine. So it yields a positive result of old rose. Then we have your Burette test for the presence of peptide bonds. So the positive result would be blue-violet. And we have your xanthoproteic test or nitric acid test. So this is for a test for substitute rings of tyrosine and tryptophan. So the positive result of this will be yellow to orange color. More tests would be Acri-Rosenheim test, so it produces violet color. Buret's test again, pinkish and violet color. Coagulation test, so it's the positive result would be turbidity or white precipitate. Then we have Forforol's test. The positive result would be deep red color. We also have Hopkins School test. It produced bluish violet corner at the junction of the sample. We also have Liverman's test. It produced fine violet blue color. Mandel's test, it produced precipitation or precipitates. Then we have your Milon's test. It produced red color, positive red color. And we also have Molish test. The positive result would be violet color. Then we have Mulder's test. The positive result would be yellow in color. Okay, so those are the different tests for your proteins. So we're done with your proteins. So this will be just an additional information. Okay. So let's discuss your blood. Okay. So when we talk about blood, this is the circulatory fluid of the body. So it is viscous and slight alkaline fluid. So what does the blood do? So to convey, they convey food materials to body cells and remove waste products from them. So they are the ones that carries oxygen for oxidation of foodstuffs and remove carbon dioxide to resulting from such oxidation. Okay, because you're again in your blood, your RBCs or your red blood cells are the ones that carries oxygen for the all throughout your body, and also it removes carbon dioxide. Okay, once it goes out to the heart, okay, it becomes a deoxygenated blood. Okay, it has your carbon dioxide already that will be released by the body. Next, it transports internal secretion or hormones and vitamins for control of the activities of the body. Of course, because your blood is the circulating fluid, okay? So it carries hormones, it carries vitamins and minerals that is needed by the body. Also aids in maintaining proper pH, water content, and temperatures of tissue because your blood also contains water, right? So the transport of water also depends to it, which controls the pH and the temperature of the whole body. Then it assists in the body's defense against disease by means of antitoxins or leukocytes or your white blood cells, right? Because your white blood cells are part of your blood, wherein these are the ones that fight the pathogens. When we say pathogens, again, these are disease-causing bacteria or agents that enters the body. So if there's an increased number of leukocytes or white blood cells, meaning there is a pathogen inside your body and they are fighting it. So your leukocytes are what do you call this? Your leukocytes are produced in a more number compared to the normal ones when there is a pathogen inside the body. Also, it transports metabolic waste to the kidneys, lungs, skin, and intestines for removal or for secretion. Okay, so those are the different functions of your blood. So now let's go to the constituent of your blood. So first we have your formed elements. So formed elements, these are your erythrocytes, which is your red blood cells, leukocytes or white blood cells, and your thrombocytes, which are your blood platelets. So when we say red blood cells again, these are the ones that carries oxygen from the lungs to the cells. And also they are the ones that remove carbon dioxide from the cell and carry it to the lungs. Okay, so that would be your deoxygenated um, blood. But when it carries oxygen, it is your oxygenated blood. So also your erythrocytes contains hemoglobin. And also your red blood cells are formed in the bone marrow. Then we have your leukocytes or your white blood cells. So it aids in the body defense against bacteria or your defense pathogens, right? So act as a phagocytes wherein it engulfs and destroys harmful microorganisms. So the way these leukocytes fight organisms or pathogens, they engulf them, okay? They eat them and then destroy them. So if WBC count rises sharply in acute infection, okay, because if you have infection, okay, your white BC, if you do a blood test, your white BC is at a greater number. Okay, the count of your RBC is greater compared to the normal ones because again, they produce they will be produced once the body knows that there is a pathogen inside. So they are the ones fighting that pathogens. And also in leukemia, okay, when we say leukemia, it's a disease, right? So if there is your white blood cells is too much, okay, it will increase too much when you have leukemia or patients that have leukemia, the YBC, the WBC count is uh the WBC count has an excessive increase in its count. Okay, so that's for leukemia. Next, we have your thrombocytes, okay, or your blood platelets. So it functions in blood coagulation. So these are the ones that to prevent, to stop, or to prevent, um, what do you call this? To prevent blood loss. Okay, so these thrombocytes prevent blood loss. So it is important in blood coagulation. So it contains cephalin, which is a phospholipid involved in the early stage of clotting. Also, your blood contains serum. So when a blood clots, the form elements or corpuscles are enmeshed in the thread-like mass of insoluble fibrin. So the clot, on standing, exudes serum. So a clear yellow liquid. This is your serum, and thus plasma without fibrinogen. So this is what you see when you have a wound, okay? And if before the blood comes out, there's a serum that coming out. So this is the yellow one, the clear yellow liquid. Especially when the wound is about to heal. Also serum is present, correct? So that's your serum. Next, we have your plasma. So this is a clear fluid portion of the body. So it comprises 55% of the volume of the blood. So it contains 92% of water and 8% of solid. So we have different plasma proteins present in your blood. So first, we have your serum albumin. It is essential in maintaining normal osmotic pressure and therefore the water balance of the body because without serum albumin, okay, your cells cannot perform osmotic pressure because when we say osmotic pressure, this is the way of the cell, okay, that um, what do you call this? That um, wherein it maintains the water, okay, the water flow in and out of the cell. Okay, so that's your osmotic pressure. So this is a property of your cell membranes. Okay, so it regulates the water to come in and come out of the cell. So that's why it maintains the water balance of the body. So a deficiency of your serum albumin causes excessive passage of water from the blood vessels into the surrounding of tissue. And this produce your edema. Okay? When we say edema, there's a swelling part of your body. It's too big because there is too much water in it. Okay? So there's an excessive water that is in the veins. So without serum albumin, this water won't come out. Next, we have your serum globulin. So it consists of alpha, beta, and gamma globulin. So gamma globulin contains most of the antibodies and antitoxins the body uses to combat infection. So this one also helps the WBC to fight infection. Then we have your fibrinogen. So it is converted, it is concerned with the clotting of blood. So later on we'll be discussing the coagulation of blood. Next. So let's discuss now your hemoglobin. So your hemoglobin made up of four molecules of iron containing heme as a red pigment conjugated with one molecule of protein globin, which is a protein histone. So this is similar to chlorophyll, which produce bluish green pigment in plants. For hemoglobin, um, it contains iron, okay, while your chlorophyll contains magnesium. So heme composed of four pyrrole rings. So its mother substance is porphyrin. So each hemoglobin is colored purple when not exposed to air. Okay, so the color of your blood is purple when not exposed to air. So we have here, so as you can see, this is a quaternary structure of proteins, right? So we have four pyrrole rings here. Okay, so this is the structure of your hemoglobin. This is the structural formula. Okay, as you can see, this is the four pyrrole ring. When we say pyrrole ring, it's a um five-membered ring, and one vertex is nitrogen. Okay, so this is a pyrrole ring, four pyrrole ring. So that's your hemoglobin. So we have different derivatives of your hemoglobin. We have your oxyhemoglobin or HbO2. So you can see HbO2, it is ferrous heme plus carbon monoxide globulin or CO globulin. So the color of this is scarlet red, and this one is unstable. We also have your carboxyhemoglobin. When we say oxyhemoglobin, this is your oxygenated blood. Okay. When we say carboxy hemoglobin, this is your deoxygenated blood, the ones that carries um, carbon dioxide. Okay, during circulation of your blood, I should say. So um, we have here your ferrous heme plus carbon monoxide becomes carboxyhemoglobin. Okay, so it is bluish red in color and more stable than oxyhemoglobin. Then we have your methemoglobin. So we have here your ferrous heme and ferricyanide. Okay, ferric cyanide. So it yields methemoglobin. So the color of this is brown. So those are the derivatives of your hemoglobin. So one disease related to your hemoglobin is your anemia. So it is a condition in which the number of red blood cells or the amount of hemoglobin is below normal. So we have different types of anemia, right? So it may result of any of the following. So it may cause hemorrhage, also deficiency of iron. So iron deficiency anemia and nutritional anemia is this one. Disease related to deficiency of folic acid, we have your macrocytic anemia of infancy and pregnancy. Then for lack of intrinsic factor or deficiency of vitamin B12, resulting in lower activity of blood producing tissues, This is your pernicious anemia. Then for anemia that there is an abnormal destruction of bone marrow by chemicals, drugs, or radiation, it is your aplastic anemia. Then abnormal hemoglobin, such as those of sickle cell anemia and thalassemia. Again, your sickle cell anemia is a mutation, correct? Point mutation. So now let's go to the coagulation of blood. So the generally accepted theory of blood clotting mechanism is as follows. So the enzyme thrombin is formed from its inactive form, which is your prothrombin. So you need thromboplastin and calcium ions to activate the active form of your prothrombin, which is your thrombin, sorry. So your thromboplastin is a cephalin, okay, a protein substance released from the thrombocytes in injured tissues, tissue cells when is shed. Okay, so once your prothrombin, with the aid of your thromboplastin and calcium, okay, forms its active form to thrombin. So this thrombin will react to your fibrinogen. So this your fibrinogen will be converted to fibrin with the help of your thrombin. Okay, so your fibrin is the thread-like, okay, structure that forms clot, okay, during clotting of your wounds. Okay, so that's the mechanism, blood clotting mechanism as follows. Okay, so again, prothrombin formed its active form, which is your thrombin, and this thrombin enzyme will react to fibrinogen to form fibrin or to form the clot in your wounds. Okay, so that's your coagulation of blood. So um, okay, so that ends our discussion regarding your proteins in your blood. So if you have any questions regarding the discussion, just feel free to message me. Okay, so I'll end this recorded discussion. Thank you for listening and have a nice day.